When we think about movement in something like bacteria, we sort of assume that it's just random movement. And if you have ever thought about this, and maybe you haven't explored this in your mind, it's because they don't have brains, right? So you're like, well, they don't have a brain, so they must just like move or flop around in the water, just float around. They're kind of a victim of whatever's happening. Or if they are moving, they're just kind of moving wherever. But in reality, in many cases, movement is very directional. So they're intentionally moving towards food, for example, or something desirable, or moving away from something undesirable. And so these can be based on cell communication, so some kind of signal in the environment that their cell is able to receive. So it's not a brain functioning to tell them where to go, but a chemical reaction between a signal and a receptor that can cause them to move one direction or another. So they have lots of different ways they move. So they can swim, they might twitch or glide through materials, or they might just be moving by adjusting flotation. We'll focus here more on the swimming and gliding. So this is a diagram showing how bacteria can move. So it's kind of interesting because they do have a certain amount of randomness to how they move, and this is what's called the tumble. So what they'll do is, say this is a bacteria. Oops, I'm in the wrong spot. It's kind of moving, and then it kind of spins around. That's the tumble. And then it starts moving this way. It tumbles around, and then it moves this way, and it tumbles around, and then it moves that way. It tumbles around. It moves that way. It tumbles around. It moves that way. It tumbles around. If you followed each individual step, it might kind of look random. But if you consider the start point and the end point, the bacteria is overall heading this direction. And so that can be in response to some kind of food source. So if there's no food source, the bacteria just kind of tumbles every which way and it doesn't care where it's going. But if there's a difference in food, then it's going to head towards that food. So again, this is demonstrating that even without a brain, they are able to move towards desirable things like food. So one of the ways that they have swimming movement is that they have flagella. So flagella are these structures that allow for a swimming movement. And so you probably saw flagella on eukaryotic cells in your previous biology class. And while we use the same term in eukaryotes and prokaryotes, they're very structurally different. So they're not made out of microtubules. So I'm just going to put here built differently. And the different components and these different building blocks cause a different type of movement. So without getting into the details, the way that a flagella moves in bacteria is like a rotor motion. So think like a helicopter, the top of a helicopter. So this is going to spin around like this. So if I use my wrist here, it's doing, oops, trying to get the camera there. It's spinning like my wrist is spinning around. Flagella move like this in eukaryotes. So in eukaryotic flagella, they get an undulation. In prokaryotes, bacterial flagella, they do this rotor movement, okay? Still getting movement from this long tail-like looking type structure, which is why we give them the same name. Another sort of interesting difference in bacteria is how diverse their flagella are. In eukaryotes, we saw, for example, a sperm with one long back flagella, or we saw a cell with two shorter flagella that it could use for forward motion. So we have these two forward motion flagella, or we have the back like on a sperm. Positioning of flagella and number of flagella in bacteria is far more variable. So in these bacteria, we see this one little flagella on one side. This is an example of a bacteria that has many flagella coming off all over its cell, like all over the different parts of its cell. Some bacteria, we saw E. coli earlier, just has a bunch of flagella coming off of its off of one side. So a lot of diversity in number and position of flagella. So what we also see in bacteria are structures called pili. And I just want to be really explicit here. These are not cilia. So back when you learned about eukaryotes, you learned about cilia and flagella as structures that facilitate movement in eukaryotes. And cilia and flagella are structurally similar in eukaryotes. So let me write that here. And they generally serve the same function, which is movement. Okay, but you may remember, again, there's my sperm cell with a flagella. What many students remember is how cilia covered the surface of a eukaryotic cell like this, and they were short. And so when you see this picture of pili, it's not surprising that you would remember cilia that kind of looked like this. But pili are actually totally structurally different from flagella in prokaryotes. So that's one difference. In addition, they have diverse functions. They're not just for motion. So in some cases, they do help a cell move across a surface. So they can sort of adhere to slimy stuff and glide. So that's one function. But they also have a sort of alternative, like opposite function, which is for adhesion. So in some cases, they actually help them adhere to surfaces. And a third function you may remember is sex pili, which are involved in conjugation, a type of genetic exchange, horizontal genetic exchange in bacteria. So depending on the pili, there's very diverse functions and structures that come under this name. So it's kind of a generic name for these thin thread-like structures that can come off of the cell surface.